Dr. Jonathan Parr. Dr. Parr is an assistant professor in the Division of Infectious Diseases at the University of North Carolina at Chapel Hill. His research interests include malaria and the infectious diseases of poverty, including recent translation projects focused on malaria parasites with false negative rapid test results due to mutations of the pfhrp2 and 3 genes, in addition to studies of viral hepatitis in the Democratic Republic of Congo at the request of the Ministry of Health. Thank you.
Hi, thanks Kumar for the introduction. I will say up front that my talk's going to be a little bit more technical in nature and perhaps boring to many of you, but the reason for the talk is that I do think the devil's in the details when it comes to testing for these deletions. Brief disclosures, I am working with Abbott Diagnostics on diagnostic development projects unrelated to malaria, but there's no financial conflict of interest there, no other disclosures. So a brief map of this talk. First, I want to get into the details of PCR-based testing for HRP2 and HRP3 deletions. Then I'll touch briefly on some alternative approaches, some of which have already been highlighted and others actually presented here at ASTMH this week. And then finally, I'll give an update on some of our work in the Democratic Republic of the Congo. So this is a slide I recycled from our HRP2 session last year, and the point of this is just to say that the lab methods to confirm deletions are complex and challenging. And the reason for this is that it's very difficult to prove the absence of a gene using PCR because there are many possible reasons for a negative result, as Jane already highlighted. A negative PCR result could mean that you have a gene deletion, but it's also possible that there's some problem with your target. Maybe you don't have enough DNA in the sample. Maybe there are mutations in the primer binding sites. Maybe you have an issue with your reagents. The list goes on and on. And so in our case, we somewhat naively jumped into this and thought, well, we'll pull some published assays and use our sample banks and generate some prevalence estimates. But pretty quickly, we realized that our simple testing algorithm needed to be revised. And by the end of our JID paper that came out this year, we ended up with a very complex and honestly convoluted testing algorithm. So again, I want to highlight what Jane has already mentioned. There are very useful guidelines that I think anyone considering this type of work should reference, especially the paper by Chen Cheng and others that I've highlighted here from the Malaria Journal in 2014. There are other guidelines available as well. Despite the existence of these guidelines and increasing interest in improving the quality of these surveys, however, when you survey the literature, you see that there are a variety of different PCR-based methods employed. So in our lab, honestly, for our own internal purposes, about a year ago, we decided we would just pull down a handful of PCR assays, and in this case, we could find 6 published PCR assays for HRP2 and HRP3. Olivia Anderson in the top corner here did most of this lab work. These included 4 targeting HRP2, 2 targeting HRP3. And to cut to the chase, there were a few interesting findings. One, at least under our lab conditions, we found that atypical PCR conditions improved assay sensitivity. Additionally, using the published assays, we found a range of limits of detection in our hands, ranging from 0.4 parasites per microliter all the way up to 4,000 parasites per microliter. So regarding atypical PCR conditions, it's our practice in our lab when we are running a new PCR, whether we've designed it ourselves or we've pulled it from the published literature, to optimize the assay. For us, that typically means that we vary the annealing temperature, we may tweak the number of cycles, maybe the primer concentration, maybe you'd even throw in some magnesium, standard molecular biology techniques. However, it hasn't been our practice to vary the extension temperature of assays. But for this particular project, in addition to comparing the limits of detection of these 6 assays, we did vary the extension temperature, and we had an interesting finding. So the gels on the right here, it's hard for me to highlight these really from here, but you can see that we used 3 different extension temperatures ranging from 60, which is a lower extension temperature than what most labs typically employ, all the way up to 72 degrees, which is a fairly common extension temperature for most available tacks. We used serially diluted 3D7 parasites, and these are just 2 example assays, one targeting exon 1, 2 of HRP2, another targeting exon 2. And the takeaway point is that as we lowered the extension temperature, the sensitivity of these assays improved. And this was true for 5 of the 6 assays we tested. Either our limit of detection dropped by approximately a log, or we got much darker bands. One possible reason for this is that HRP2 and HRP3 are AT-rich targets, and it's possible that the DNA melts at the higher extension temperatures of 72 degrees, which makes it challenging for the polymerase to extend along the template. This is not a novel finding. I'll say that there's a great paper from Dr. Wellham's lab from the mid-1990s where they published an elegant brief report looking at a variety of falciparum targets, specifically long fragments, and found that for multiple targets, by lowering the extension temperature, they improved their sensitivity. I also mentioned that we were interested in determining the limits of detection of these assays. The takeaway point for us was that not all assays were equal. Again, these are 2 example assays shown on the right. Again, looking at HRP2, exon 1, 2, and exon 2. And just to highlight exactly how these gels are set up, again, we ran 3 different extension temperatures, and we did serial dilutions at each temperature. You can see that there's a big difference in performance between this assay on the top and the assay on the bottom, approximately 100-fold difference in sensitivity. So why does this matter? Unlike most PCR assays that we employ in our labs where we're looking for a positive result, in this case, we really care about negative results. And if you're employing an assay with a high limit of detection, it's quite possible that you can misclassify a negative result as a deletion. Another interesting finding, after we had completed the limits of detection testing and compared the effect of different extension temperatures, we applied these assays to several common lab strains. DD2 DNA, which is a common lab strain that originated from Southeast Asia, is known to have an HRP2 deletion. When we ran our exon 1, 2 assays, and to be clear, these are assays that primarily amplify exon 1, but they go through the intron and also catch a little bit of exon 2. There we go. To our surprise, when we tested a known HRP2 deleted strain, we got bands. On the bottom, I have 3D7 DNA, which is known to be HRP2 positive. So my initial thought with this was, well, this is just a classic example of contamination. We'll just repeat all of our assays using different stocks. But we actually repeated this result, and we found something similar using our HRP3 assays against HB3 strain DNA, which Dr. Wellham's already mentioned before, which is known to have an HRP3 deletion. So we then took this product and just simply sequenced it. This is a figure showing an alignment of HRP2 and HRP3, including the first exon and some of the intron. As one of Dr. Wellham's figures emphasized, these genes are very similar in their sequences. When we sequenced the products from the gel I showed on the previous slide, the top one, the DD2 product with a known HRP2 deletion, actually I've got them switched here, but 3D7 with an intact HRP2, we found that in fact the DD2 product was HRP3. And obviously it's hard for you to see the bases probably from the back of the room, but I've just circled regions of homology between our DD2 product and HRP3. And so our takeaway was, at least under our lab conditions with the HRP2 exon 1, 2 assays, it was possible to amplify the paralogous gene. This is important because depending on the sample type that you're working with or the parasites in a particular region, you may actually misclassify parasites as wild type if they have, for example, an intact HRP3 gene, but they lack an HRP2 gene, as will be reported in the paper that came out, I guess, today or yesterday from Kenya. And the opposite is also true. If you were doing an HRP3 assay and you had an HRP3 deleted parasite, but a parasite with an intact HRP2 gene, you could misclassify the sample. And just to emphasize what I said before, we probably should have done this in the first place, but this is just simply an alignment of HRP2 and HRP3. For the 6 assays that we tested, we simply mapped the primer binding sites. And if you look closely at the primer binding sites, especially those for exon 1, you see that there's high sequence homology between the 2 genes. And so it's quite conceivable that you could amplify one or the other depending on your conditions. So for us, having done this initial testing, I'll go back to my initial slide. Our motivation for doing this was simply to improve our own lab workflow and to really drill down and get to the bottom of what our bans or lack of bans meant. And at present, this is our lab workflow. There are probably multiple ways that this could be done, but our current approach is that we are performing a quantitative PCR step first and defining our parasitemia. In order to proceed to further testing, we want to see that the sample has at least 10 times, sometimes 100 times more DNA than the limit of detection of the downstream assays. This is a conservative adjustment that has some problems when you're thinking about generalizing this to your analysis, but I do think will give us some protection against the risk of false deletion calls. Then we proceed with testing for HRP2 and HRP3 in parallel. And we have now shifted to testing exon 2 only simply for 2 reasons. One, it improves our lab workflow, and secondly, it helps us avoid the risk of spurious amplification of the paralogous gene. Finally, if we have a candidate deletion, then we follow it up with a confirmatory PCR. In our case, we use a separate real-time assay that has limited detection, also 10-fold higher than the HRP2 and HRP3 assays. So now I'd like to talk briefly about alternative approaches. So hopefully Kumar will touch on this a little bit in our question and answer session, but there's a paper in PLOS ONE that came out this year from the CDC group by Eric Raget and John Barnwell describing their Luminex platform, which has been successful in detecting very low levels of HRP2 and has the potential to allow high throughput streamlined testing that's not at risk of some of the molecular issues I've just described. There was also a poster from Alfredo Mayor's group, poster 293 yesterday, describing a Luminex-based approach. And I know that we've got Christian Kupli here in the crowd. He had a very nice poster yesterday describing a new digital droplet PCR assay that could also open up some opportunities for high throughput screening. Having talked a little bit about the PCR issues and the difficulty of proving that a deletion is there, there's an obvious appeal to using next generation sequencing approaches. However, these approaches are not free from some of these same issues. Using short read next generation sequencing technologies, you still have to deal with the problem of aligning these reads to a highly repetitive region involving genes that have high homology. So in our experience, this does create some problems when you're attempting your alignments. Additionally, even though you could generate some decent alignments and use the eyeball test to say there seems to be a deletion or not, we really need strong objective approaches to identify deletions. And as with many whole genome sequencing analyses, this really depends upon good sequencing depth, which can be challenging if you're working with dried blood spot samples, for example. So finally, I want to just show 2 slides to highlight some of the work that's ongoing in the Congo. Jane highlighted our paper that came out in JID this year. We performed a large survey of asymptomatic children in the Democratic Republic of the Congo and reported a prevalence of approximately 6% HRP2 deletions. We used a microsatellite panel developed by Kumar's lab to do some basic population genetic analysis and found that the deleted parasites were genetically distinct from controls. However, one thing that Jane mentioned in her talk that I do think is important to highlight again is that sample selection is extremely important when you start to think about policy implications. In our case, through our collaboration with the DRC Ministry of Health, we've had the opportunity to perform large surveys using their samples collected during DHS surveys. These are obtained from asymptomatic subjects. When you think about policy changes, what we really care about is making sure that patients who are sick with malaria get the right test. And so, in collaboration with our partners at the Kinshasa School of Public Health, SANRU, and the National Malaria Control Program, and with financial support from the Global Fund, there is a survey now in 3 regions, including Kinshasa, Kivu, and in the north, Bawele, looking at symptomatic subjects only. And I will say anecdotally from our own experience in the Congo, I'm curious to see what we will find. I haven't seen a lot of signal to suggest to me that we're missing many symptomatic cases. And so I do think from a scientific standpoint, there's some interesting questions to ask about why that might be, and hopefully we'll have some relevant answers for policymakers soon, but also some interesting opportunities for hypothesis generation. So in conclusion, the molecular confirmation of these deletions is very challenging. Careful laboratory protocols are required. There are multiple alternative approaches under development or recently published that could have some nice utility. And there are new studies underway in the DRC, but also in Ethiopia, Sudan, and elsewhere, as Jane highlighted. Brief acknowledgments. I want to thank Steve Meshnick and John Giuliano, my primary mentors at UNC, as well as our collaborators in the DRC at the Kinshasa School of Public Health, SANRU, and the National Malaria Control Program. And then thanks also to the ASTMH and the Thrasher Research Fund for funding. That's all. Thanks.